Well, thank you very much, Dr. Sakas, for this wonderful talk. Seguimos agora com a nossa próxima palestra, com a doutora Stine Kristensen. Dr. Kristen is the head of the Laboratory of Reproductive Biology at the University of Copenhagen, Denmark. Please, Dr. Kristen.
Thank you so much. Thank you. So it's always difficult to follow Danny with all of his fantastic pictures, images, and videos, but I will give it my best try. So thank you very much to the organizing committee. This is not the first image. Can I please have the first slide? This is in the middle of presentation, so please provide the first. Yes, thank you. So thank you very much for both Aline and Mauricio for inviting me. It's fantastic to be here and to be part of this very impressive program. So the title of my talk is the in vitro culture of follicles. And this is not something that we have come up with in recent years. This has been going on for more than 4 decades now. So one of the first milestones in the culture of follicles was actually back in 1989. And this was John Epic, who actually produced the very first little mouse pup from these in vitro grown oocytes. And he actually named it Egbert. So that was a cute little name, and it was a very happy little mouse. But it actually developed into being obese and diabetic, and it died quite early. So it just shows that this system it wasn't perfect from the beginning, but not much is, I think. But it has advanced a lot since, and throughout the 90s, there was a lot of healthy mice pups born from these in vitro grown follicles and oocytes, and it does show that it can be done in mice. But it's not as easy in humans, and this is why I have this subtitle of my talk, because we're actually going into a much more complex system when we're talking about human follicles, and I'll talk about how we're now trying to bioengineer these artificial ovaries. Okay. So with that, then the primary goal of this follicle culture is to actually utilize this extremely huge reserve of ovarian follicles that we are born with. So it is these tiny follicles, the early stage follicles, the primordial and preantral stage follicles that we are targeting here. And I think what has always amazed me about the system and the ovary is that we are born with this huge amount of follicles from the beginning, a huge surplus. And it's actually really difficult for me to explain why we have this, and that's a big mystery. We have a huge surplus, we have no stem cells like in the testis, we just have one big pool of follicles that we utilize throughout life. But it's a very ineffective system because there's a huge loss of these follicles throughout life. We're born with around 1 million follicles in each ovary at birth. Around puberty, we have around 400 left, and then we only utilize 400 of them. So only 400 make it to the mature oocytes that you normally see in the clinic. So why is this? Why do we have this huge waste of follicles? What are they there for? But that's really the potential here, that we do have access to them. They're there. But how do we get hold of it? How can we actually overcome this system and make them useful for the women? That's what this research is all about. So we know it's not because they're genetically set up wrong, because you know from the clinic, you can do the stimulation, you can still rescue some of these antral follicles. So they are healthy, they're good. They're just not getting through the normal little loop in natural selection, but they're there. So how do we utilize a lot more of this ovarian reserve? So tomorrow, Michelle will have a talk about the in vitro maturation of these antral stage follicles. But today, we'll talk about utilizing these tiny, tiny preantral stage follicles. It's quite a different system because you have to remember that normally this development takes 3 months. So it's extremely complex that we now want to try and mimic this in vitro. But there is huge therapeutic potential here because there's so many of them, and for especially the human system, we know very little about what's controlling the activation and growth of these early stage follicles. So just figuring out the mechanisms regulating and recruiting the follicles is a big part of it. But also, as also Dr. Burton said about this whole model system for toxicology screening and drug testing, this is also part of the potential. And then you can say in the long run, of course, if we can make these follicles or oocytes available for the women, then you suddenly have a huge pool of follicles and potential oocytes available. So even for the patients in the clinic, this is very, this could be important. And then for the final point, this is actually the women that I normally see in our clinic because we do fertility preservation. And for these women, sometimes they have these blood-borne diseases or diseases in the ovary where we cannot secure that there's no malignant cells in the tissue. So for these patients, we would also need to develop these systems to avoid retransplanting malignant cells. So there's different groups of patients for this. And this is just how it looks in my clinic. We received an ovary. I'll talk more about this tomorrow, about oncofertility. But here, when we transplant the tissue back, there might be a risk of these malignant cells. And studies from Brussels have shown that we can isolate, you can see in the bottom panel here, that when we isolate the follicles, then we don't see these disease genes from patients with leukemia. So it is possible to isolate the follicles and put them into culture, and then we could potentially restore fertility in these young leukemic patients. So that was more the clinical aspects. So again, this has been actually in research for a long time, 40 years, and a lot of it has been in rodents. So of course the mice, as you can see, a big part, 44% of the studies. So it's all in mice. And what we just know already is that there's a long way from mice to humans, because in mice, this follicle development takes around 3 weeks, a little less. In humans, it's 3 months. So it's a lot different, this system. And that's what we're kind of up against, because we have tested a lot of different systems. There's both the 2D systems and the 3D systems. Also, as Dr. Burton talked about, the 3D systems are a lot more attractive because that's mimicking the physiological needs that the body we are in 3D, not in 2D. And it's been tested in a lot of different species, even in a large domestic animals. But it's just very variable how well it actually works. But what have attracted a lot of attention is these hydrogels, the extracellular matrix gels that you can do. You can have them both as a coating or in a structure, 3D structure. You can also have these suspension cultures that we call matrix-free. So this is where you can have it in a hanging drop or in these magnetic beads system or rotating systems. So there's a lot of different ways as how you can culture these. But again, the results are just very varying, and you can see it reviewed in some of these new review from Helen Picton. But how do we select the best for the human follicles? And for a long time, we have been using what's called alginate. It's a hydrogel from brown algae, so it's natural. It's very flexible. It has big pores, so we can have both FSH and other hormones and nutrients going in and out of the gel really efficiently. And this is the first study from the group in Chicago, where they were actually able to grow these isolated follicles. And as you can see, just to the right, you can see this is the alginate bead. And right inside, there's a little red dot, that's the isolated follicles. So this is how it looked, very low practical. But you can then culture these follicles for up to 30 days. And here you can see how it has actually developed an antrum in the end, from this early stage follicles to an antrum. It actually grew to 1.5 centimeters. So this is huge. And the matrix was actually able to sustain this growth. And you can see here also the hormones, how the follicle actually secrete the hormones that we would expect with the estradiols and inhibins. So again, it's doable, but this is again 1 out of hundreds of follicles which were grown. So the efficiency is very low. What you can see here is that when we do this culture, we would also need to do the in vitro maturation in the end, where we actually put in the HCG. And this is this pictures from our lab with my PhD student, Stine. She's very good in the lab, just to show you the whole course of the culture. So in the end, when we have this big follicle with the antrum, then we open it up very gently, and then we put it into the IVM medium for these 36 hours, and then we can retrieve the oocytes. So the first study which actually came out and showed that they had a mature oocyte after this follicle culture was the group again in Chicago in 2015. And they cultured the follicles in this little alginate bead. And they were able to show that they actually got this mature oocyte. And you can see the spindle is organized nicely, and then it's organized around the chromosomes. And they were also able to show that the follicles that developed these mature oocytes, the cumulus cells also expressed these cumulus expansion markers. So it was the first sign that the system can work with these human follicles. The next studies, there's only 3 studies which has been able to do this successfully, and they all use different culture system. So this second study is from 2018 from Evelyn Telfer's group in Edinburgh, and they use this multi-step culture system. And they start by culturing the follicles in the cortex, and then after 8 days, they isolate these secondary stage follicles with little needles, put them into a culture, and then in the end, they take out the cumulus oocyte complexes and do the IVM. And they have actually also been able to grow some of these follicles to the stage where they had mature oocytes. But as you can see, this is not really healthy oocytes. You can see there's huge polar bodies. So it could sustain the growth, but the quality is not there for the oocytes. And that's what we see. The efficiency rate for this, how well it succeeds, is very low. It's a few in hundreds of follicles, and then the quality is compromised. So the new study, they used no matrix, but they then did some AMH modulation, and then they actually did a 6 weeks culture period, which is longer than the other studies. So this is double up on time. So they did it slower, and they were actually able to, you can see again, these antrum formation in the follicles, and they were able to get these M2 oocytes that look more normal than what has been shown previously. So that's a very latest study from 2021. So that's how far we are right now with the culture systems, showing that we can do it, but it's not very efficient. And that's what I'm writing here, that we can see it's not very efficient. So it seems a little random sometimes that we do get these M2 oocytes. And it seems like we are missing something. And this is where I always get a little excited about these ovaries, because they are extremely complex and they're so dynamic, and they, like, throughout the whole reproduction and the development, that the follicle starts outwards, growing inwards, going out again. It's a very dynamic organ, even through the menstrual cycle. So to think that we can just simply put it into a little gel and culture it, that seems maybe a bit too optimistic. So I think we really do need to go in and look at the whole environment of the follicle, the extracellular matrix that is supporting the growth of these follicles. This is what we did a little commentary on, and that I think we cannot just say that one little follicle can do it by itself. It's used to having neighboring follicles. It's used to being affected by growing follicles, late-stage follicles. It's very dynamic, and we have to try and mimic this system a lot better. So that's where we're going into the little more complex bioengineering, which has been shown in animal studies to be beneficial for this development. So we'll have a look at that now. So we're talking about this artificial ovary, but more it's a bioengineered ovary. And there's different matrices, different scaffolds. The fibrin is one which is used in a lot from groups in our field of fertility preservation. It has been supporting follicle growth quite well. There's also the 3D printing. That's more one of the new approaches from Monica Laranda in Northwestern, where they actually 3D print this gelatin ink to make these little constructs of ovaries that they can actually transplant back to the mice and actually provide ovarian function in mice. But a lot of things is doable in mice, so it's not that easy just to put it into human settings. But it's just proof of concept that this is ways that you can culture follicles. One of the newer approaches is also how can we mimic this normal ovarian extracellular matrix. And you can see here to your left there's a human cortex, how it looks, and then how you can try and mimic that with these matrices that is being developed. So there's a lot of work going into mimicking these matrices. But what we came out with, and other people came out with, why not just use the matrix we already have in the ovary? Why try to mimic it? Why not just use it? Because we have the ovary and extracellular matrix, and that's what they did in this study. This is a bovine ovary. They have removed all the cells, so it's only the matrix left, and then they put on these ovarian cells, and then they're actually able to reconstruct these little ovaries, transplant back to mice, and induce puberty in the mice. So you can actually just use the native matrix from the ovary. So that's also what we tried to do recently. So we have access to this human tissue. So we took some human tissue, put some mild detergent on it, so you remove all the cells. So you're just left with this pale white matrix. You can see it's in picture B. So you just have the matrix left, and then you recellularize with these isolated follicles. So we did that, and then we transplanted it to mice. And after 3 weeks, we retrieved the grafts, and were able to show that some of these follicles were actually able to survive in this decellularized tissue. So that's just another approach, how to try and mimic the ovary in different ways. So with that said, then I have to go into the future, with, which also Dr. Burton touched about, because this is the microfluidics. How do we combine all these cell types in a more complex formation? And that's what we can't really do with the more simple systems, with the decellularized tissue or hydrogels. It's not complex enough, because we're not able to mimic the real dynamics in the ovary. We can little bit mimic that with these microfluidics, because we can control what's going in and what's coming out. So this is a paper which is actually the same as Dr. Burton showed you. Yet you have these little chambers and little capillaries where you can control what's going in. And here in this study, they mimicked the FSH and the HCG peak. So they cultured the follicles and looked how they responded. And here it was mouse ovaries also. So here you can just mimic the whole menstrual cycle. And then you can look how follicle development progress. And they were actually able to do that and show that these follicles were able to secrete the estradiol, and then after ovulation, they secreted the progesterone, and they actually turned into corpus luteums. So these microfluidic systems, they're quite fancy, and I think this is where we need to go, because it's really trying to make a much more complex system work together when you have the different cell types in the same setting. And one of the newer papers, just again, this is just so amazing with the ovary. It's so complex, because you have the hard cortex, soft medulla, and the follicles grow in and out in these different compartments. So that what they have done here is they have made these little gels, which are soft in the middle and then more stiff on the outside, trying to mimic the ovaries and putting them into these microfluidic systems. So this is, again, we're getting a step closer to what a normal ovary would be like for these follicles. And I think it is the future. And talking about the future, then I have to go into this, because maybe we won't need these follicles. Maybe we'll just make them from stem cells. So that's the new thing, that maybe this will be all abundant, because we can just take, in this study from Hikabe, they took the tail tip of a mouse, extracted, they reprogrammed them in the iPS cells into these primordial germ cell-like cells, and then they constructed these little organoids or ovaries, and then they were able to actually grow these in vitro and fertilize them and put them back into the mouse, and then they had the offspring, and they could even do the cycle again with the embryos. So they could do the whole follicular genesis and fertilization and embryogenesis in vitro in these mice from some somatic cells. So maybe we won't really need all of these eggs. We'll just make them from stem cells one day. But I think that's when I get very, very old and die. But maybe but maybe for multiple systems, we can still use it. So this is what we are working on now with a group in our university, making these ovarian organoids. That's an add-on to what Dr. Burden was also talking about, the organoids. They are very attractive at the moment. And again, for these in vitro systems, for drug testing, everything. So with that said, what does the future hold for this follicle culture of the earliest stages? We have made a significant advance in the last decades. We are getting there, but not as much with the human ones yet, but we're still getting there. And it is encouraging that we can make these meet phase 2 oocytes, even though they might not be completely competent or of high quality. We're getting there. But still, a lot of challenges remain. And when we look into the next steps, we really do need to secure that we develop these more complex bioengineered systems by the new bioengineering, which is very hot at the moment, and that we get these healthy oocytes that can actually be competent and give some offspring. But with that said, I just want to thank my fantastic colleagues in Copenhagen, and thank you very much for your attention.